So just to remind you, the working hypothesis of the lab, so what I'm going to tell you about today is unpublished data. The new data I'll show you are unpublished, and it really presents what we think may be a useful model for how we think about administering drugs that might have broad effects on senescent cells. And then I'm going to end by showing you new data to suggest that there may be mechanisms for which this secretory phenotype was selected that has positive effects for the organism. So just to remind you, the model that we're working on is the idea that the mechanisms that protect us from cancer for the large part of our lifespan, half of our lifespan, has a trade-off, and that trade-off are phenotypes that we recognize as aging. And those trade-offs are really carried out by these two tumor suppressor functions, or these two tumor suppressive cell fates. And the one we're focusing on mostly is this idea of cellular senescence, this permanent arrest of cell proliferation, which is accompanied by this secretory phenotype that's pro-inflammatory. So the model is that in a young tissue where you have very few senescent cells, with time, with age, cells undergo senescence, and those can be epithelial cells or stromal cells or any cell type that has the ability to divide. These cells, of course, are protected from becoming cancerous, so they can't undergo cell proliferation, so they'll never form a tumor. But as a consequence of this senescence response, they begin secreting a large number of extracellular proteins, inflammatory cytokines, proteases, growth factors, and that secretory phenotype then disrupts the local tissue structure and therefore tissue function, and that would cause the degenerative changes that we associate with aging. And if there happens to be a pre-malignant cell in the vicinity of this disrupted local tissue, then the tissue constraints on that pre-malignant cell are now released, and the cell can then go on and actually form a tumor. So we would predict that these cells are responsible for both degenerative changes associated with aging as well as late life cancer. So I introduced to you the idea that once we understood some of the pathways that drive this pro-inflammatory secretory phenotype that we think is driving aging, and I described for you the DNA damage response pathway. I alluded to the fact that the mTOR pathway was also involved, and this P38 MAP kinase NF-kappa B driven pathway, that by understanding these pathways, we have the potential to intervene with this inflammatory phenotype and hopefully break this link to age-related disease. So I alluded to the fact that the mTOR pathway, which you heard this morning from Misha Blagoensklale, is really important and drives a number of very important aging phenotypes, may be key to understanding this inflammation. And so I'm going to pick up on that and just remind you that the mTOR pathway is probably the most ancient, ancient and conserved pathway that we know of that drives aging. So high TOR, just to remind you what Misha told us this morning, high TOR drives aging, and if you can back off on TOR activity, suppress TOR activity, you can ameliorate some age-related diseases. So the work I'm going to tell you about today was largely the work of Remy Martin-Labarge, Art Orhalo, and I really have to give credit to my co-worker at the Buck Institute, Pankaj Kapahi, who works on TOR in flies and convinced us to do these experiments in human cells. So just to remind you about TOR, there are two TOR complexes that are formed by the mammalian TOR kinase, and they're called TORC1 and TORC2. And TORC1 is really the target of rapamycin. So of the two complexes that are formed, rapamycin selectively targets the mTORC1 complex, and mTORC1 is a regulator of translation. So it primarily regulates both cap-dependent and cap-independent translation, depending upon which arm it's regulating. But this, just bear in mind, is primarily a pathway that regulates mRNA translation. So here's the first experiment that Pankaj inspired us to do, which is to take these are senescent human cells in culture. This is looking at IL-6, pro-inflammatory cytokine, And as I told you yesterday, pre-senescent cells secrete very little of this cytokine. Senescent cells secrete a lot. And if we now titrate in, this is a dose response to rapamycin, you can see that rapamycin causes a marked suppression of IL-6 secretion. Now, there are a couple of things to note about this dose response. The first is there's a rapamycin-sensitive component to this secretion, and that is about 80% of the secretion is under TORC1 control, so it's inhibited by rapamycin. And then there's a TORC1 independent component. So this is not complete absolute suppression of this inflammatory cytokine. The second point to note is that we get maximal suppression at extremely low doses of rapamycin. So this is a 0.5 nanomolar, and it's basically maxed out as far as its suppression. So by and large, we tend to use maybe between 2 and 10 nanomolar for the rest of the experiments I'll tell you about. But just bear in mind that these are very low doses of rapamycin. In fact, these doses are even lower than the doses that were found in the blood of those mice who had the lifespan extension due to rapamycin administration. So one of the first things we wanted to do was we wanted to know whether this suppression of IL-6 was peculiar to IL-6 or whether this whole 40 cytokines that are expressed by senescent cells are also affected by rapamycin. So here is an antibody array. For those of you who may remember, these are these membranes with 120 different antibodies labeled on them. And then what we do is we take conditioned medium from cells, in this case either pre-senescent cells, either treated with the vehicle, which is DMSO, or treated with rapamycin, or senescent cells. In this case, we induce senescence with exoradiation. And they secrete lots of stuff. So we've rank ordered the degree to which they're secreting these 40 or so cytokines. And these are the cells now that were treated for 7 days with rapamycin. And the important point about this array that it told us about was, first of all, there were some cytokines that were very strongly suppressed by rapamycin. But there were also cytokines that were completely unaffected by rapamycin. So again, what this tells us is that by inhibiting TORC1, we're suppressing some components of this secretory phenotype, but not others. And we think that this might be important later on when I'll talk to you about some of the side effects that we might expect from administering rapamycin. Okay, so if rapamycin is suppressing the secretory phenotype, which we think is driving degenerative changes of aging as well as late-life cancer, that's good. But of course it would be a disaster if rapamycin undid the growth arrest, because it's the growth arrest that suppresses cancer. So this is an experiment that shows that rapamycin does not affect the growth arrest. So here is BRDU incorporation. Pre-senescent cells incorporate a lot. So this is a measure of DNA synthesis. Senescent cells incorporate very little BRDU. And in the presence of rapamycin, that DNA synthesis index remains very low. So that's good. It says that we're not overcoming the growth arrest, and we shouldn't be driving cancer by treating animals or people with rapamycin. So this was all well and good, but very soon after beginning to explore the mechanism by which rapamycin do this suppression of the secretory phenotype, we ran into two puzzles, which took us a while to figure out. So the first puzzle is this. This is again IL-6 secretion. Pre-senescent cells secrete very little. Senescent cells secrete a lot. And what we found is that we could add rapamycin for 1 day after we induce senescence, in this case, again, by ionizing radiation. And we assay now for IL-6 production 7 days later. And we get as much suppression as if we had rapamycin present for the whole 7 days. So this made us begin to think that, well, first of all, what these data say is that rapamycin might be acting at a very early step in the development of the secretory phenotype. So just to remind you, I told you yesterday that this phenotype takes a few days to develop. And here we are getting maximum suppression before we even get full development of the phenotype. And the second point, the second possibility that it raised is that there might be some positive feedback loops that are being broken by inhibiting the TORC1 complex. The second puzzle came when we began to look at mRNA levels. So I told you TORC1 primarily regulates translation. And so we expected the mRNA levels of most of these inflammatory cytokines to be unaffected. And very much to our surprise, we found rather large decreases in the mRNA levels of several of these inflammatory cytokines. And again, this puzzled us until we checked one more cytokine. And that cytokine was IL-1 alpha. So IL-1 alpha is an unusual, it's a very prominent pro-inflammatory cytokine. It's unusual because it's not secreted, so we never see this cytokine in our arrays. It's a membrane-bound cytokine that binds its receptor within the same membrane of the same cell. So this has been called juxtacrine stimulation. And it's one of the first cytokines that is expressed in the development of the SASP. So what happens is IL-1 alpha binds its receptor, and that receptor binding sets off a signaling cascade that ultimately activates the transcription factor NF-kappa B. And it turns out that NF-kappa B is a very potent inducer of IL-1 alpha gene expression. So what we realized is we were dealing with this positive feedback loop that we had reported over the past couple of years, and this is just a summary of our published data. So we know that one of the first cytokines to respond to senescence stimuli is IL-1 alpha. It activates NF-kappa B. NF-kappa B in turn activates more IL-1 alpha, and this positive feedback loop begins to be established. Eventually, this microRNA cluster is induced, which tends to tune it down so it doesn't get out of control. But meanwhile, you build up enough NF-kappa B activity to begin driving the transcription of IL-6, IL-8, and all of those other pro-inflammatory cytokine genes. And so if this is correct, that this feedback loop is the target of rapamycin, it makes some very specific predictions. And so we began to test those predictions. So the first prediction is that we should be able to overcome the effects of rapamycin by providing cells with exogenous IL-1 alpha, recombinant IL-1 alpha. And that's exactly what we've done here. So here's IL-6 production, rapamycin suppresses it, but now we come in with recombinant IL-1 alpha. We can completely overcome the effects of rapamycin. The second prediction is that rapamycin then should target the translation of IL-1 alpha mRNA as opposed to the transcription, which it doesn't. So these are polysome gradient profiles. And the way to read these profiles is that this region of the gradient are messages that are untranslated, and this region of the gradient are messages that are translated. And we're comparing now the translation of different mRNAs in either pre-senescent cells, or these are the two most important comparisons, senescent cells either treated with a control or treated with rapamycin. So basically we're comparing the red and the green. So here's tubulin, which is unaffected by senescence and is unaffected by rapamycin. So its position in the gradient doesn't change. And here is IL-1 alpha. And you can see in senescent cells, most of the IL-1 alpha mRNA is in the translated portion of the gradient. Whereas in the presence of rapamycin, there's a marked shift now to the untranslated portion of the gradient. And here is one of those cytokines that is unaffected by rapamycin. When I showed you that antibody array, I showed you some of these cytokines were not affected. And so here's one of them, MCP4, and you can see now the red and the green are nearly on top of each other. So indeed, its translation is not targeted by rapamycin. And then finally, this makes a very firm prediction. If it's true that rapamycin acts by breaking a positive feedback loop, it should have effects that last well beyond the period at which we administer the drug. So I told you yesterday that the presence of senescent cells can stimulate the growth of tumor cells in a xenograft model, in a mouse xenograft. And so what we've done in this experiment is we've taken one of those xenograft models. These are breast cancer cells, human breast cancer cells growing in a nude mouse. And they grow with a certain trajectory. They've been stimulated now by the presence of senescent cells. So over the course of about 6 weeks, this is tumor growth. In this experiment, we took those senescent fibroblasts and we pre-treated them with rapamycin ex vivo in the culture dish for 7 days. So the animals and the tumors are not seeing the drug. And now we inject them into the nude mouse, and we very much attenuate tumor growth. So this result actually really gave me hope because, as I mentioned yesterday, there's really no such thing as a perfectly safe drug. And I think for the purposes, if we're thinking about drugs that can improve longevity by suppressing these pro-aging pathways like the mTOR pathway, we want to avoid constant chronic administration of a drug, just on basic principles. And this kind of result suggests that it may be possible not to have to chronically take such a drug, but to take something intermittently or in a pulsatile manner that can break positive feedback loops. And for sure, eventually, we know actually, eventually the cells will overcome this inhibition, and after several weeks, the tumors begin to grow again. But the point is that we're optimistic that we may be able to use this principle to develop therapies that don't require constant administration of a drug. So I'm going to leave that story now as a little bit of hope that there might be some pharmaceutical interventions. And now I'm going to switch gears and try to address a question that has really always plagued me. And I've never really given, even to myself, a satisfying answer. And the question is, why on earth do we have this senescent phenotype? We have apoptosis. It's a perfectly good tumor suppressive response. It has none of these complicated secretory phenotypes that drives inflammation and causes trouble later in life. And why did nature need to put into place this senescence response? So one possibility, which I've never quite bought into, but it's a possibility, is it's so important to suppress cancer, you want a backup mechanism. That's possible. But I'm going to show you data now that suggests that the senescence response was probably under positive evolutionary selection because it has a role, a positive role, in wound healing. So let me show you these data. So the old perspective is that senescent cells are bad, and I still believe that. I think all of the data that I showed, which suggests that these cells can drive aging phenotypes, are correct, and there's even more evidence mounting, especially from the group at Mayo Clinic. But there is now this new perspective that we're considering, which is that there are some benefits to having a senescence response above and beyond the fact that it suppresses tumorigenesis. So as you may recall, I alluded to this paper from the Mayo Clinic in which my colleagues Jan van Dersen and Jim Kirkland made a transgenic mouse in which it was possible to eliminate senescent cells at will. So we made a parallel mouse, and I'll explain to you how this mouse works. What we did is this is a transgene that is on a huge piece of DNA, a bacterial artificial chromosome. And what we've done is we've taken a senescence responsive promoter, in this case, the promoter for the P16 gene. This is a tumor suppressor, and it is induced under many, if not most, conditions in which we induce senescence. So it's induced whether it's oncogene-induced senescence or telomere-induced senescence, DNA damage-induced senescence. This gene is activated. So this promoter is very responsive to senescent stimuli. And what we've done is we've engineered the P16 gene in this bacterial artificial chromosome so that it no longer drives P16, but instead it drives this protein called 3-MR. And 3-MR stands for trimodal reporter. What it is it's a fusion protein between luciferase, red fluorescent protein, and the herpes simplex virus thymidine kinase. So this viral thymidine kinase has a very high affinity for this drug, ganciclovir. And what it does is it phosphorylates ganciclovir. When ganciclovir is phosphorylated, it gets incorporated into DNA by DNA polymerases, and it terminates DNA synthesis. And it turns out from experiments that we did to validate this system that the major recipient of phosphorylated ganciclovir is not nuclear DNA synthesis, although it does get incorporated into nuclear DNA synthesis, but mitochondrial DNA synthesis. So even non-dividing cells are killed by ganciclovir because, in the case of non-dividing cells, because of toxicity to mitochondrial DNA. That causes the cells to undergo apoptosis and they die. There is a cellular thymidine kinase. Fortunately, it has a very low affinity for this drug. So this is more or less specific to cells that express the viral thymidine kinase. So with this transgenic mouse, what it allows us to do is to monitor the appearance of senescent cells using either luciferase or red fluorescence, and inducibly kill the cells in vivo by giving the animals ganciclovir. So the first thing we did was to use a skin wounding protocol. And I'm going to show you now data that suggests that senescent cells are very rapidly induced at the site of a wound in the skin. So that's shown here. So here are two transgenic animals. Here's a wild-type animal. And this is 4 days after a small 6-millimeter punch biopsy. And what you can see is this bioluminescence, which is indicative of the transgene being activated as a consequence of the wound. And we've now taken these wounds and we can do RT-PCR, and we can show that there's P16 expressed, the 3-MR transgene is expressed, and IL-6, this pro-inflammatory cytokine, is expressed. And the other part of the model is to be able to eliminate these cells now with ganciclovir, and that seems to work very well as well. So here is a 3-MR mouse that's been given just the vehicle, and here are two 3-MR mice that have been given ganciclovir prior to wounding, and they never developed the bioluminescence, they never expressed P16, 3-MR, or IL-6. So the system seems to be working. We can take senescent cells, we can show that they appear very rapidly at the site of a wound. We can show that these senescent cells are cleared from the wound very rapidly by ganciclovir. And just parenthetically, if we don't treat the animals with ganciclovir, the senescent cells are eliminated naturally in the granulation tissue, the scab that comes off at the site of the wound. So that would be the body's natural way of eliminating those cells. So now the question is, what happens to wound healing if we prevent senescent cells from appearing at the site of the wound? Just to remind you about wound healing. So wound healing, this is true whether it's a cutaneous wound in the skin, but it's true for almost any kind of tissue damage that occurs in vivo. Those types of trauma, in order to heal, usually require 3 phases. And the first phase is an obligatory inflammatory phase. Following that inflammatory phase, there's a phase that's followed by cell proliferation, and then finally there's this matrix formation and remodeling of the wound. So if this doesn't occur, wound healing doesn't initiate properly. If this doesn't occur, the wounds don't close properly. And if this doesn't occur, the wounds remain fibrotic. So all 3 phases are needed for proper wound healing. So what happens when we eliminate senescent cells? So probably this is the best panel to focus on. So here are wild-type animals, wounded and treated with ganciclovir. They close their wounds in about 5 days. These are the animals where we eliminated senescent cells at the site of the wound by treating them with ganciclovir. And you can see that they take about twice as long for the wounds to heal. And when we excise those wounds and look at the quality of wound healing, these animals heal with much more fibrosis than these heal. So these data then suggest that there may be a positive benefit to having this senescence response, and that is to really promote wound healing. Okay, so those of you who are not asleep from your lunch, you probably are scratching your head and saying, wait a minute. We know that senescent cells accumulate with age, And we also know that wounds certainly don't heal any faster as we get older. In fact, that's one of the hallmarks of aging skin, is wound healing slows down. So how can we reconcile this idea that you need senescence to promote wound healing, and yet older skin, which has more senescent cells, actually heal wounds less well? And to answer this question, we move to a different mouse model. And this is a mouse model that has been done in collaboration with Simon Meloff, my colleague at the Buck Institute. What Simon has done is he's taken a mouse and he has made a floxed allele of the SOD2 gene. So SOD2 is the superoxide dismutase. That's the mitochondrial specific enzyme. So this is the major enzyme for detoxifying oxidative, reactive oxygen species within the mitochondria. So we can take his mice and we cross them to a Cree that would excise this gene, and that Cree is driven by a keratinocyte-specific promoter. So we're only going to eliminate SOD2 in the skin, in the keratinocytes. And the Cree is actually a fusion to a mutant estrogen receptor that keeps the Cree inactive until we give the animals tamoxifen, which then allows the Cree to become active. So the system seems to be working well. We can eliminate senescent cells specifically in the skin when we do this cross. And when we look at skin that is devoid of SOD2, we see the appearance of senescent cells within the skin. The difference is these senescent cells are persistent. They last and they are not eliminated, so far as we can tell, or at least not eliminated very quickly. So what happens to wound healing now in these animals where senescent cells are persistent? They're no longer being eliminated in the granulation tissue. And that's shown here. So here's a control animal, and here's an animal treated with tamoxifen. So again, wound healing is retarded. And this has sort of made us think then that really the positive effects of senescence are very time and place specific. So they're induced certainly at the site of a wound. They need to be present for a short period of time, and then they probably need to be cleared in order to resolve perhaps the inflammation that they're driving. And if they're persistent, you really have the same problem as if they were completely missing. So this sort of has left us now with a new view of how we view the senescence response, which is that it really is a balance between positive effects, obviously suppressing the development of cancer, which we still think is the primary goal for instituting this growth arrest, but also now participating in a positive way in tissue repair. And balanced against that are, of course, the bad things that I told you about yesterday that senescent cells do. They can disrupt normal tissue structure and function. They can drive age-related cancer. But they can also now be responsible for retarding tissue regeneration and repair if their presence is persistent within tissues. And so again, when we think now about trying to intervene in this very complex process that has both positive and negative effects, I think we have to think much more multidimensionally. We have to think about intervening intermittently at the right time at the right place. For example, you would never want to take rapamycin if it suppressed the development of a secretory phenotype in senescent cells before you were having surgery, something like that. So I will stop here now and again thank all of the people in the lab who contributed to this work, and past lab members with whom we still collaborate, and really a great group of outside collaborators. And I should point out this mouse was made in collaboration with Jan Huimaker's lab in Erasmus University. So thank you for your patience.
Exciting new view of senescence. So I'm wondering, and maybe ignorant about it, so the signaling pathways that trigger senescence in case of tissue repair and wound healing, are they similar to what they used to talking about?
Yeah.
Or pathways and something?
Yeah, so it's a great question, Vera, and we just don't know the answer yet. So we're doing those experiments now to see if we inhibit, for example, ATM, MAP kinase and all of that if we also interrupt wound healing, and we just don't have the answer. But it's, yeah.
So you point out the selective advantage of some points, and the bad things, you talk the side effects. The side effects, you assuming the aging is not programmed. Aging.
Programmed, you mean in the sense of being evolutionarily programmed? I mean, this is a program, right? This is a program, but it didn't evolve for the purpose of making us old and decrepit.
Yes, but suppose aging somehow is beneficial for...
Oh, it is bad. I can attest to that.
For bad for?
For me.
Yes, but in the evolutionary perspective, all that trade-off and the benefits and the side effects could be beneficial for this species, for evolution. If you have all this being beneficial, in an ad libitum program, in ad libitum aging, you would have all this as a beneficial effect. You don't need to assume it's a bad thing. Any of that experiment results, any of it is bad if you assume aging is programmed, is desire for evolution.
Yeah, so I would like anyone who believes in programmed aging to explain to me how, if you have two alleles within a population, one causes you to be decrepit and one allows you not to be decrepit, how you select for the allele that allows you to be decrepit, especially post-reproductively.
Yes. So you connect it with reproduction, and then if you have a mutation who you inhibit this problem also deactivates reproduction. It cannot be selected in an ad libitum situation.
Maybe we can talk about this after, because this is going to be a debate.
Really a focus always. I wonder if senescent cells are likely to be the recruiters of somebody else at the site of wounding. Have you looked at the content of the invaders to the wound in the presence and the absence of senescent cells, and how does it change?
Yeah, so it's another great question, and we're doing those experiments right now. I mean, I wish I had answers. So this is very new data, and so we know that they recruit natural killer cells. Scott Lowe has shown that. And so we would expect there to be those wounds to be devoid of NK cells. We're just staining the tissues now.
Did you try to track the origin of cells which cover the bone? Whether this is stem cells, tissue stem cells, vocal stem cells, or circulating cells?
So the wound recruits, as Andre suggested, immune cells. It also does recruit stem cells, in this case from the skin. But the source of those cells that become senescent, we think, are mostly myofibroblasts. So we think they're the ones that are initiating the response. And then we don't know which of those factors is responsible for the recruitment.
Judy, you focused on wound healing, which is predominantly a skin phenomenon that in the model that you use. But of course, fibroblasts contribute to the connective tissue of many of our soft tissues, and the HSVTK transgenic mouse allows you to actually look for beneficial effects of senescent fibroblasts in other tissues. Have you done that?
We're starting. So I can tell you a tiny, tiny piece of preliminary data. So we're working with somebody who works in a stroke model. So apparently, when there's a stroke, there's also this obligatory inflammatory phase that's required before the infarct is walled off. And so all I can tell you is that we do see bioluminescence at the site of the stroke, bioluminescence. So we think senescent cells are being produced very early on in the stroke, and we're just looking now to see what happens when we eliminate those cells. So again, early days, sorry.